Assalamu alaikum, dear students. Last time we discussed the regulation of gene expression, and it can be best explained with the help of Operon system in prokaryotes, and this gene regulation of gene expression or these operons may have more than one or more than one structural genes: an operator gene, a promoter gene, a regulator gene. Besides, there may be a repressor, an inducer or a co-repressor, that is from outside. Now this operons on the basis of regulation of gene expression can be of two types, that is, one is called as inducible, another is called as repressible operon, or you can say inducible operon, or the regulation of gene expression can be inducible or repressible. Now first, the inducible, it is a regulation of gene expression. It is a type of regulation of gene expression. Regulation of gene expression or a type of operon, which is switched on, that is, it becomes functional in response to the presence of substrate, and it occurs in catabolic pathways. That is why there is breakdown, or you can say hydrolysis of substances, that is called as catabolic pathways. And there is an example we have to discuss later on it, that is Lac Operon. I have already given you the concept of this Lac Operon. There is another system, that is repressible system, or repressible operon, or repressible regulation of gene expression. Now it is also a regulation of gene expression. Or regulation of gene expression or a type of operon in which the product of gene activity if already present stops the activity of said genes, stops the activity of said genes, and it occurs in anabolic pathways. That is where there occurs the synthesis of substances, that is anabolic pathways. And there is an example of this, that is called as tryptophan operon. Tryptophan operon Tryptophan operon is an example of repressible system. Now, where there is no regulation, there is no regulation of the gene expression, that is called as constitutive. Constitutive regulation of gene expression. Where the regulation is absent, the genes are hence active or their enzymes are always operational throughout the developmental stages. That means there is no regulation in this, that is switched on or switched off. And we can besides have the operons may have a negative control or may have a negative control or positive control. The operons may have a positive control or negative control. Now, negative control, in the negative control, the regulatory product जो है, the regulatory gene product, the regulatory gene product, that is the product of regulatory gene that shuts off or switches off the expression of genes. Switches off the expression of genes. Switches off the expression of genes under its control. Under its control. And the positive control is, it is negative control. Negative control. In the negative control, the regulatory gene product shuts off the expression of genes under its control. And here is tryptophan operon. Now what it means, इसका मतलब क्या है? अगर एक ऑपरॉन है यहाँ पर, इस ऑपरॉन में स्ट्रक्चरल जीन्स हैं ABCDE, इसमें एक ऑपरेटर जीन है, इसमें एक प्रमोटर जीन है और इसमें एक रेगुलेटरी जीन है I. जब ये ट्रांसक्रिप्शन करेंगे, ये स्ट्रक्चरल जीन्स, दीज आर स्ट्रक्चरल जीन्स, जब ये ट्रांसक्रिप्शन करेंगे, आप मान लो ये ऑपरॉन जो है ये स्विच्ड ऑन है, it is switched on and it forms the enzymes 1, 2, 3, 4, 5, 6. Enzymes 1, 2, 3, 4, 5, 6, and then these enzymes जो हैं, they may help in the synthesis of tryptophan. This tryptophan जो है, it acts as the regulator. This tryptophan then acts as the part of the repressor, and this repressor then shuts off this gets attached to operator gene, and that may control the promoter gene, and there is no functioning of structural gene. That means it switches off. It indirectly switches off this. This is the gene product. This gene product shuts off the expression of these structural genes. It is said to be negative control. Negative control. Whereas there is positive control. There is positive control. In a positive control, the product of regulatory gene, product of regulatory gene activates the expression of genes. Activates the expression of genes under its control. That means the substrate or the product of regulatory gene that helps in switching on the structural genes, and there is an example that is called as Lac Operon. And in case of Lac Operon, there is inducible system. So it is an inducible and it has the positive control. So Lac Operon जो है, it has the positive control and it is an inducible operon. Now last time I said that there was the Jacob and monad, F Jacob and J monad. In 1961, they put forth the concept of operon, and one of the inducible operons is Lac Operon. And this Lac Operon consists of various genes. Lac Operon जो है, इसका structure जो है, it consists of various genes. There is first one, the structural genes. There are 3 structural genes: Z, Y, and E gene. There is an operator gene. There is a promoter gene. And there is a regulator gene or I gene. Now these structural genes जो हैं, these genes actually synthesize mRNAs. The structural genes actually synthesize mRNAs. That is, cistrons responsible for transcription of polycistronic mRNA. As it is found in bacteria E. coli. The Lac Operon of E. coli contains now the three structural genes. There is Z gene, there is Y gene, and there is A gene. Now this Z gene codes for the synthesis of enzyme, codes for synthesis of enzyme lactose, codes for the synthesis of enzyme lactose, or it is also called as beta galactosidase, beta galactosidase, or it is shortly called as beta gal enzyme. Beta gal enzyme. It is primarily hydrolyzing or breakdown, hydrolyzing or breaking lactose into glucose and galactose. Glucose and galactose. The Z gene जो है, it synthesize the enzyme lactose or beta galactosidase. This beta galactosidase is used by this bacteria for hydrolyzing lactose into glucose and galactose. Then these substances जो हैं, glucose and galactose, they act as a source of energy for the bacteria. So there is another structural gene. This is Y gene. There is then Y gene. Y gene synthesizes, it codes for the synthesis of enzyme permease. It codes for the synthesis of enzyme, that is permease. Permease increases the permeability of the cell. It increases the permeability of cell to lactose. इसका मतलब अगर लैक्टोज मीडियम में है, उसको बैक्टीरिया के अंदर जाना है. Now when the enzyme permease is synthesized, it increases the permeability of the cell membranes to make the entry for the substrate, that is lactose. And there is third one, the A gene, another structural gene, that is this one. This codes for the synthesis of enzyme trans-acetylase. Enzyme trans-acetylase. This codes for the enzyme trans-acetylase, or it is also called as thiogalactosidase. Thiogalactoside acetylase. And you can remember the only one name, that is trans-acetylase. This trans-acetylase enzyme जो है, it is used for metabolizing or neutralizing toxic thiogalactosides. Thiogalactosides. It is used for metabolizing toxic thiogalactosides, or you can say the neutralizing the toxic substances within the cell. Now, these substances which are allowed by enzyme permease. जैसे परमिएस एंजाइम परमिएबिलिटी बढ़ाता है मेम्ब्रेन का, तो लैक्टोज के साथ कुछ टॉक्सिक सब्सटेंसेस भी अंदर जा सकते हैं। तो उन टॉक्सिक सब्सटेंसेस को न्यूट्रलाइज करने के लिए ये और एक एंजाइम बनाता है, दैट इज ट्रांस एसिटाइलेज। और ये ट्रांस एसिटाइलेज मेटाबोलाइज करता है या न्यूट्रलाइज करता है टॉक्सिक सब्सटेंसेस को। Now these three enzymes are produced in different molar concentrations and become active or inactive simultaneously as per the requirements. I may revise these three enzymes जो हैं, they are produced in different molar concentrations and become active or inactive simultaneously. And there is another that is called as the operator gene, that is this one. This operator gene जो है, it controls the synthesis of mRNA. Operator gene जो है, it controls the synthesis of mRNA over structural genes. Synthesis of mRNA over the structural genes. Over the structural genes. It is switched off. It is switched off. It is switched off by a repressor. By a repressor. By a repressor. It is switched off by a repressor. एक रिप्रेसर से ये स्विच ऑफ हो सकता है. So if a repressor gets attached to operator gene, it may not for synthesize the mRNA, and which activates the Z gene, Y gene and A gene. So अगर एक रिप्रेसर होगा इसमें मीडियम में, तो वो स्विच ऑफ करेगा इस सारे को. पहले वो इनएक्टिव करेगा ऑपरेटर जीन को. एन इंड्यूसर, इंस्टेड ऑफ एक इंड्यूसर जो है, एन इंड्यूसर, इसमें वो है लैक्टोज. एन इंड्यूसर जो है कैन इनएक्टिवेट द रिप्रेसर, वो इनएक्टिवेट कर सकता है, इनएक्टिवेट द रिप्रेसर, इनएक्टिवेट द रिप्रेसर and switch on the operator gene, and switch on the operator gene, switch on the operator gene. Then operator gene directs the, then operator gene जो है, it directs the structural genes to transcribe their respective mRNAs, transcribe their respective mRNAs. And it is revealed that this operator gene जो है, it is made up of only 27 base pairs. ये बना हुआ होता है 27 base pairs का. And it has palindromic sequences. Palindromic sequences, that is repeated sequences. Repeated sequences in the reverse direction, जो last time मैंने आपको बताया था. For example, if there is G, C, A, C, G, there is T, there is T. This is called as palindromic sequence. That is T, G, C, A, C, G, T. In reverse, it is the same. T, G, C, A, C, G, T. It is called as palindromic sequence, which is same in the reverse direction. It is called as palindromic sequence.